commands that are commonly used in Linux, like to move the file, to create a file, to move from one directory to another. Those are like basic commands that we'll generally use here. Then we'll move on to this command line navigations. These are the commands by which you can like efficiently master the files, like moving from here and there. And then there will be certain commands also that will be given to you to perform the analysis, like fastqc, that are supposed to be written on the terminal. So when you write a command on terminal, like a command line on terminal, then it is executed and then you can get a result. So that and all also we'll understand today. Then we'll be also looking at data management, that is how well you work with complex sequences. And the main part about today's lecture is we are going to install certain things, like we are going to install certain tools like fastqc, sam tools, hiset, and there are certain other tools in our terminal environment. And then we are going to install R and R Studio for our Windows. Like I'm considering that most of you are working in Windows, right? Like you have a basic exposure of R, sorry, not R, Linux. Like a few commands will be done in Linux, and most of the commands that we run in R, we'll directly run it in R Studio, because R Studio is compatible with Windows. Like you can run it in terminal as well, but here we'll be running it in Windows. Like we'll download a GUI and we'll work with that. So based on that, it will be easier for you also, you guys also, to catch up, because we like it will be like an app. So you open the app, you run the commands, and you get the results. So this installation followed by the packages. So once you install R, then there are certain packages and libraries that needs to be installed that is going to help us with the analysis. So today, basically, we are going to prep our systems for the further analysis, where we'll install all the tools that are important. We'll see and we'll learn the basic Linux and the basic command lines that are important to work with Linux. So why are we using Linux for bioinformatics? So most of the tools that we are using, that whatever we are going to use, like these tools are just specific for our RNA-seq, like fastqc and trimming and mapping. Like these are common ones. These can be used in whatever analysis you want to do. So whenever we do any analysis, say ChIP-seq, ATAC-seq, DNA assembly, genome assembly, RNA-seq, the first step is that understanding the quality of the data. So fastqc is generally run in terminal, and that remains constant for all the analysis that we're going to do. Because as we move, the first step that we take is to understand the quality of the data that we have received from our sequencing machine. So all these tools, whichever we are going to use, they're highly compatible with Linux, and bioinformatics heavily relies on Linux because of these tools itself. So all these tools that are used, most of them are made so that they can be used in the Linux environment. That is why we preferably choose Linux whenever we have to do a bioinformatic analysis. So modern sequencing technologies can generate like hundreds and gigabytes or terabytes of data, and processing these all information requires a little more efficient computer systems. So Linux is what it provides. So Linux provides us with this resource meant like this high computational power. It uses the RAM, storage, and the stuff to basically understand and operates the system. So Linux is an operating system where you are able to like more efficiently work with, because it is very much compatible with the work that we are trying to do. It supports automation scripts. That is, if you have multiple samples, say 100 of samples, so you don't have to run the command 100 times. You can write a script. After you run that script once, all those commands will be run 100 times, because you have put it in a loop in the particular script. So all these automations are very much possible in Linux, and therefore you prefer this particular environment. Compatibility, if we have to go. So Linux is more compatible as compared to other systems. Therefore, it is necessary that we generally use Linux for our advanced bioinformatic research. Essential HPC and cloud computing tools for computing itself, and most NGS tools are Linux based. So as I just mentioned the name of certain tools, all those tools are generally based on Linux. So you can run them directly on command lines when you do it in research. So I'm constantly using this word command line. So command line is basically the codes that you write when you work in Linux. So I'll just show you once. So when you open terminal, so you get a pop-up. So screen like this. This is my PowerShell for my Windows. So you guys will be familiar with this when you switch to the Linux environment. So now I am shifting basically by writing WSL. It is my Windows subsystem Linux. So I have installed Linux inside my Windows. So I'm going to switch my system now from my PowerShell, that is my Windows, to my Linux system. So in that Linux system, we'll be working and we'll be writing all those commands. So whatever I have, so now you can see that this is my Linux system. So generally, this is what the prompt
Anshu,
looks
sorry
like.
to
So
interrupt you.
here you write whatever you have
Miss
to write.
Anshu,
So you
can you
can see
hear
that
me?
I'm writing something.
Sorry to interrupt you.
Yeah,
Before
yeah.
proceeding, shall we check with our participants, those who are going to work along with you?
Yes, Pavani, I couldn't get you. Can you please come again?
Before you work on commands, shall we confirm with our participants who are ready to work along with you so that it will be easier for us to check with them, right? What do you
Yeah,
say?
yeah, yeah. I'm just showing a demonstration right now. So whenever I'll move on to the working part, I'll ask all of them to have their systems open so that they can also work
Okay,
like side by side.
all right, fine.
Okay.
Please continue.
So whenever you open a Linux window, it generally looks like this. So you have a prompt. So from here, after the dollar sign, you have to write whatever commands you have to execute. So whatever you write here, this basically acts as your command line. So this is your prompt that you give, and that is getting executed, and you're getting your files that are generated by that particular command. So generally, we'll work on the command line prompts. That is, we are going to do all the analysis like fast QC, the trimming, mapping, all of that as a command line comments. So that I'll teach you in my further analysis in the further slides when we move. So now the major concept that you need to understand is that Linux, why are we using Linux? Because most of the tools that we are using is highly compatible with Linux, and Linux provides a very good environment for us to study even the larger sets of data. Like if you have very large data set like gigabytes and terabytes, you can use that as well and understand here. So in RNA-seq, what we are doing here is we are just implying and we are analyzing the RNA, that is the transcriptome samples by using the Linux commands and shell scripting. So shell scripting I just mentioned, right? When we have this, when we automate a pipeline, that is, instead of writing each command again and again on the terminal, what we are doing is we are trying to create a script where we are putting a loop. That is, that command should be run for 100 samples, but you are not going to run it. The system is going to run it. That is known as shell scripting. So all these features are favored by Linux. That is why Linux is favored by bioinformaticians to do their analysis. So now I would like to summarize the NGS pipeline that we have that we discussed yesterday. So when we have an NGS pipeline, so from today we'll only discuss after the fast QC files. So yesterday we also discussed how we are generating the RNA-seq data, but now that is not much important to us. So from now on we'll be moving forward with how are we going to analyze the files and like what are we doing with the files. So after we do everything, so our NGS pipeline, that is our RNA-seq pipeline, starts from a FASTQ file. You have done everything, you have got the RNA sequence, and now you got the data. So from here, the data analysis steps start. So you have a FASTQ file. Okay, now this FASTQ file contains all the data that has been sequenced by the sequencer, and it has been arranged in a specific format that also we'll learn today in what format it is present. And after we got the FASTQC file, now we move to the next steps. So the first step was getting a FASTQ file. Now the second step is quality control. Quality control is going to be the first thing for everything. See, whenever you go out to buy something, a cloth, of like to eat food or something, quality is the thing that you everybody looks for. If you're buying a pant or a jeans, you check for the cloth, like is the cloth a good quality cloth? If it is not, then you generally don't go for it. But if it is, then only you move further. So similarly, whenever we have a raw data files, the first step that we follow is the quality control. After the quality control is done, we understand what is the quality of the base, what is the adapter concentration, what is the GC content, everything we understand. Then we move to the next step. Next step is alignment. So you can use HiSet2, Bowtie2, other tools are there as well to align them. And after aligning, you're getting two files here, either a SAM file or a BAM file. So the difference between SAM and BAM file is one is a file where you can see the aligned reads. That is the SAM file. Whereas the other is a BAM file. BAM file is a binary file. It is in a reduced, like this, the size of the file is reduced and it is compressed because it is a binary file, so that it is easy to store in the system. And these BAM files, they are very like these BAM files are being read by the analysis tool, whatever we if we are doing a feature count, if we are doing mapping, whatever we are doing, these BAM files are what are considered inside the environment by our tools. After we have the SAM and BAM file, next thing is we do is a sorting and indexing and aligning of these BAM and SAM files or the stats that are present. For this, the input will be a BAM file because we need to sort it. So we'll be studying about these also in a little more detail. Okay, so after we sort the and index the BAM files, then we move to the read counting. That is, we'll construct a count matrix. That is, it will have the samples and the genes. So it is going to tell the number of reads that are corresponding to those samples, and we'll get a sample count. That is a count matrix. After we get a count matrix, this count matrix acts as an input for the differential expression. So you put this count file so that now you want to understand that okay, which genes are more expressed and which genes are less expressed. So now you do the differential expression. So the tools that are used for differential expression is the seek2 and HR. So they run a series of commands. So before, like, what are the series of commands that are run? So first we input the read count matrix. Now what will happen is it is going to estimate the log to fold change, the p-value, the significance calls. I'll mention all these also, like when we are going to do it more in detail, like as we move on, when we will get the differential expression, I'll make you understand what each value is indicating, and we'll go slowly. For the time being, it might feel like jargons to you, but you just have to understand right now that what is basically happening in this step. So now you do the differential expression. So the main step that is happening here is normalization. So you bring down both the data onto a same scale so that you can compare it without any bias. Okay, so after you get that, you plot. So plotting is basically either a heat map or a volcano plot. We'll study heat map as well and volcano plot as well, how to basically interpret them. And then after that, you basically try to find the biological meaning by doing the enrichment of this biological data or the plots that you have gotten. So this is a basic workflow that we are going to follow. So we are going to do each and every step individually. We are going to understand what is happening, what files are getting generated, and how do we interpret them. So now we'll come back to the Linux operating system. So as we are using Linux, so Linux has three things. One is a shell, one is a kernel, and one is a terminal. So terminal is what I just showed you. So this is what is terminal. So terminal is the place where you are generally working, and to this particular prompt, you are giving your prompt. So now it has three parts: kernel, shell, and terminal. So kernel is basically the core of the operating system, that is Linux. Linux core is called as kernel. So kernel is basically involved in the memory management, in process scheduling. Like if you have given 100 processes, so it is going to give like numbers to them. Okay, first you will go, second you will go, third you will go. So like this it is, because if all of them go at the same time, it is going to cause some what do you say, crashing of the system. But if you give each and every process, that is what you want to do, the work number, okay, first you go and get it done, second you go and get it done. So like that, if you give a process like a proper schedule, that is known as process scheduling. And this is good and very better, like if you have to give them work in a manner, in an order. So all the work will be done simultaneously. It will not even crash. It might take some time, but that is like good, right? But the work is happening at a proper interval of time. Then hardware interaction, it communicates with the hardware. That is, hardware is the place where basically your data is getting stored in the hard disk or whatever. So that direct access from the terminal to that place is done by basically kernel. So every program has to communicate with the kernel. So whatever you're writing on terminal, it goes to the hardware after communicating with kernel. So shell is the interpreter. So shell basically acts as an interpreter between the user and the system. So kernel is a part of a system and terminal is a part of a user, because on terminal, the user writes the command. Those commands are interpreted by shell and then executed, and then the result is sent to kernel. So whatever kernel sends it back, it is again interpreted so that it can be read in a human format. That is what we are looking for. So shell acts as an intermediate between both terminal and kernel. And the terminal is just an interface that with which we interact, and whatever we write goes to the shell that interprets it and it sends to the kernel. So whatever commands we write on the terminal, those are the command line prompts, and those are interpreted by shell. So I hope this combination and the connection between kernel, shell, and terminal is clear. Is it clear, guys? Okay. So most of our commands, this is just a backend that is happening. We don't need to worry about that. We are going to be basically working on terminal. We are going to write our commands. We are going to get an output. So now we are going to understand, this is a basic concept of Linux file system. So how is a thing getting stored in a Linux system? So it is like a tree. So that is, a tree has a main branch, then sub branches are coming out of it, then again sub branches are coming out of it. So that is how it basically acts. So it acts like a hierarchical structure. So you guys know, you guys have seen a phylogenetic tree. So it is, the Linux file system is also like a phylogenetic tree. So it has a main root directory. From that root directory, we keep on forming a home directory, a subdirectory, all of that. So generally, these are the common directories that are present. That is, you have the root directory. From the root directory, we have a home directory. Then we have a user directory, a binary directory, etc., and a data directory, etc., and a data directory. So these are a constant directories that are present under the root directory. So we generally don't touch all of them because it has its own subdirectories and data. We generally work under the home directory. So we'll just understand what these directories are important for. So home directory is basically that contains our files. So whatever file that you are generating or whatever you are working with, so all those files are generally saved in the home directory. Say you created today 1.txt. So that 1.txt file is saved in your home directory. So then we have binary. So binary generally contains any essential executable programs. So whenever you are going to write like ls, means these are the commands that we are going to learn now. So any command that you're going to be executing, so those commands listed, those commands are essential, and these commands are basically executed by this binary version. Then we have the USR. So this USR stores basically all the installed softwares and libraries. So whatever you're installing an application, it goes and it gets itself installed in the user section. Then we have the etc. So etc contains all the system configuration files that are present in your system. Then TMP is the temporary files. If there's a temporary file that is present, it will go and get stored in the TMP folder. And then this TMP folder, as it's temporary, it keeps on getting cleared periodically. So whatever files are stored here, they will get cleared until and unless you want to make it permanent and shift to any other folder. And majorly the data. So data is where are all the this is all the data file, that where are all the bioinformatics data, whatever we are generating is getting stored. So you can create this file under the home section and all of that. So the major files that we generally don't touch in the Linux file system is user bin, etc, and data, because they contain certain files that is not much of important to us, but it is much important for the Linux system to function. So the major working folder for us is going to be the home directory, where we can create subdirectories and form multiple text files or whatever data we want to store. So the key, so we understood from this particular thing that in Linux file system, the Linux follows a hierarchical file system. That is, you will have a root, then from that one file is emerging, then from one again, from that one again. So it is like a ordered way. So that is how it is present. So now we'll move to the Linux commands. So I want to ask you guys, how many of you have Linux systems installed in your systems and you are able to access it right now? Can you just raise your hands?
Dear participants, hope you got Anshu's question. Those who have Linux environment installed, could you please raise your hand? Thank you, Girish. How about others? Before that, I would just want a quick confirmation whether the class till now was clear for you guys. Can I get thumbs up for that? Thank you. If yes, everyone is active, please give thumbs up those who have Linux environment, because we already spoke about this in our previous sessions itself. So I anticipate everyone have installed Linux. Those are using Windows. Yeah, please be active, guys. Thank you. And
Okay.
Ms. Usha, I guess most of them have Linux.
Okay. So whoever has the systems handy with them, so maybe they can work alongside with me. So what basically it is going to be a demonstration, and you guys can work itself, like work also along with me. So it will be like hands-on for you guys, and whoever doesn't have a Linux system in hand right now, they can just see what I'm going to show. I'm going to run each and every command, and I'm going to show like what is this command doing and what you guys can do with it. So if you have it, you guys can work according alongside with me, and if you guys have any doubt or are you not able to understand what command happened or what I did, so you guys can just stop me, I'll repeat it again. So first is this basic Linux navigation commands. That is, first is the present working directory. So what is a present working directory? So this is my terminal. So if I write the command pwd, so this command basically tells about the present working directory. So it will give me an entire path that, okay, if I have to check from the, this is my root directory, okay. From my root directory, I have a directory C, users and Anshu, and then I'm checking what is my current working directory. So when I see that, I can see from my root directory, I'm moving like from these, that is MNT, inside MNT, I have C, inside C, I have user, and inside users, I have Anshu. Okay, so these are the directories that are present, and currently I'm working in what directory? I'm working in the directory Anshu. So this is the entire path to my directory. If I want to, if somebody wants to come to my directory, like if somebody wants to come to my house, I have to tell them in the entire path, like, okay, you have to take a right, a left, and then come. Similarly, if you have to come to Anshu, you need to follow this path. So this path is important. Whenever we do some analysis and studies, we need to give the path, the entire path for a directories for the commands to run. So PWD tells us the current working directory, that is the present working directory. So the next command is ls. Ls is going to list all the files. So as this is my normal directory, like this is my directory in which I have stored a lot of stuff. So ls will show all the directories or all the text files that are present. Okay, so you can see whatever is present, you can see these are the text files. Like you might not understand, we'll create one and then we'll understand. Okay, so I have created a, you ignore that command, now you understand. So in pwd we got the present working directory. So now I shifted to this folder. So now you can see in home I have for the path from to my home, it is from this root directory, and I have to follow all this and I will reach home. Then I will see ls stands for list. It will list all the files or folders that are present in your current directory. So right now I am inside my home directory. So inside my home directory, whatever files and folders are present, if I put ls, it is going to list that. Okay, so as it is highlighted in green, it might be different in different systems. So for my system, the thing that is highlighted in green, it shows that it is a directory. Directory means it's a folder. That is, I can go inside this folder. So ls is going to list everything. If there was nothing in my current directory, if I write ls, it will not show anything because there is nothing. My current directory is empty. So whenever you have to list any file or anything inside your current directory, you use the command ls. Then there is another command, ls minus lh. It is a more detailed version of ls. That is, ls will only show the name of the directory or the file that is present. But when you do ls minus lh, it is going to show you multiple information. It is going to show you the file permission. So this is the file permission, and it is going to show you the directory under which, like the home directory, the root directory, all of that, the ownership of this file. It is going to show you the size, the day and date, the time and date on which it was created. So just apart from the file name, it is going to show multiple information of the file that might be important for somebody to like do a study, do analysis, or for checking results or anything for their own purpose. So this command ls minus lh is going to give a little more detailed version of whatever is present in the file. So now, if I have to go from home to Anshu, or maybe see, I have If you just want to clear everything, you can just write clear and everything will be done. So now I list all the files that are present in my home. So now I want to go into this folder because I have told you, right? This is a folder. So I need to change my directory from home to this folder. So I will write cd. Cd means change directory. This is another command. And then you will write the name of the folder, that is RNA-seq lecture 2. So now as I click, you can see that my folder is getting changed. Initially, I was in the folder home. After running this command, I moved to the folder RNA-seq lecture 2. So this command cd is used to change the directory. So similar way, I went from home to RNA-seq. I can also go from RNA-seq to home. So I will just write cd space dot dot. And I will move back to my previous directory. Now I'll again go in my further directory, that is RNA-seq. I'll again do cd RNA-seq. I want to see what all files are present in RNA-seq lecture 2. I will do list and I can see whatever files are present. Here you can see that this is not highlighted as the my above file. So this states that this is not a folder. This is a text file. If it is highlighted like this or it is present in a different color, that means that it is a folder. And you can go inside a folder, but you can never go inside a text file. If I try to do cd 1 plus txt, as it is a text file, I shouldn't be able to get inside it because it's a file, it's not a folder. So it is going to show an error. It will show that 1.txt is not a directory, it is a text file. That is why this command is not working. But it worked with RNA-seq lecture 2 because that was a folder that I had created inside my home directory. Is these command clear? The changing directory, listing files, these are very basic commands that you will be working with a lot when you move to the RNA-seq analysis. Is it clear? Okay. The guys who had the systems ready with them, did you guys run the commands? Were you guys able to understand whatever I just ran here? Were you able to replicate that?
Those who are working along, can you please unmute and see what's happening? Let's the session be interactive. Any volunteer from Holy Cross? Are you guys working along with Anshu?
Yes, ma'am.
Is this commands working at your end?
Yes, ma'am.
Fine, thank you.
Okay.
Anshu, let's resume the
Yeah.
session.
So these are the basic commands which were, these are the Linux navigation commands. So we understood all of them. So now next is creating and managing files. Okay. So now we'll again go back to our terminal. So you can see here. So this was a folder that I've created, right? So how have I created this folder? So when you have to create so I'll do list file. So only my one text file is present in my RNA-seq lecture 2 directory. Now I want to create a folder. So the command to create a folder is mkdir, that is make directory. So say I'm making directory as Bangalore. So I'll make mkdir Bangalore. So now in my particular directory, that is RNA-seq lecture 2, another folder is going to get created, that is Bangalore. So when I list the files again, initially it was just 1.txt. Now you can see another file that is created, that is Bangalore. Okay. So this is how you create a folder, a directory. Now, what you want to do is you want to replicate 1.txt. Say you need to copy this file. Okay. So, sorry, first we understood now how to create a directory, right? Like Bangalore. So now you should also understand how can we create a text file. So for creating a text file, we write touch and say, now I want to create 2.txt. So I'll do this. So now when you see list files, you can see that there is a 2.txt text file created and a folder named Bangalore is also created. So these are the two commands that are used for creating a folder and creating a text file. Okay. So now next is copy. So you need to copy 2.txt. You have some content in the file 2.txt. Okay. So wait a second. I'll just write something inside 2.txt. Okay. So I have written hello inside my 2.txt. So how am I going to show that? So we have a file, you can see 1.txt and 2.txt. So either we can go and open it, either we can go into our subsystem, open the file and see that, okay, hello is written, or otherwise we can display it onto this terminal as well. So the command to display whatever is present in your file is cat followed by the name of your file 2.txt. So you can see hello is printed here. So this hello is the content that is present in 2.txt. My 1.txt file is empty. If I do cat for 1.txt, you can see that nothing came because the file is empty. Okay, so that is how the cat command is used to visualize basically the 2.txt and the 1.txt, like whatever text file you have, to visualize the commands inside it or text inside it, you use the command cat. So now I want to copy this file, that is 2.txt, into a file named hello. So there's a command called as copy cp. What file you have to copy? 2.txt. Now you want to copy this file into another text file. Like you want to copy the content of 2.txt into another file. So this is my file from which I have to copy, and you need to give the name of the file. So say the new name of a text file I want to give is a dog. So now you can see that in my folder, you can see there is 1.txt, 2.txt, and a dog is also created. So when I try to visualize what content is present inside my dog, you can see it's hello. Because I copied my 2.txt file, I made a copy of it and I named the new file as dog. So cp command is used to copy. Now I don't like this name dog. I want to change the name of this file. So now I will use the command mv, that is move, and I will write the name of the file that I want to change. Now what name I should keep? Say hello is written inside, so I'll keep it as hello itself. I want to change the name of my file dog to hello. So we are going to use a command mv move. So now you can see when I list the files, now my file has changed name to hello. Now no more dog is there. So now I'll cat, like that is I'll see whatever content is present in my file. You can see hello. The text and the content in the file remain same. The thing that changes is only the name. To change the name, we use mv. To copy, we use cp. Okay, now I don't want only this hello file. Why should I make a hello file when I already have a text and a text file and a folder? So I would like to delete. So I will use rm, that is remove, and the name of the file that I want to remove. So ls. So it will remove my file. So hello is removed from the picture itself. So from these commands, you are able to understand how to create a directory, create a file, how to copy a file, how to move a file, and how to remove. Okay, so another command. So now I don't want this 2.txt to be here. I want this 2.txt to be inside the Bangalore folder. So the mv command that we use for renaming, it is also used for moving the file. So mv 2.txt. So 2.txt is the file that you want to move. Now where do you want to move it? So now you will give the entire path. So this is the entire path. I will copy. Okay. So the entire path you have to give from the root directory. After RNA-seq lecture 2 also, there is a folder Bangalore. So I want to copy it inside Bangalore. So now I want to move my file 2.txt from this folder to the Bangalore folder. So I've given the path for the Bangalore folder. Okay. So now you see, I'll do list. Now the Bangalore, the 2.txt file is gone from here, because I've moved it to Bangalore. So now I will go into Bangalore folder. How will I change the directory? By using the command cd. So I'll put cd, name of the directory in which I want to go, and then I will list the files. You can see that I have moved the file 2.txt from the RNA-seq lecture 2 to the Bangalore folder. So mv can be used to rename as well, and it can be used to move the files as well if you give a proper path where you want to move it. So this is how you move and change the files in Linux. Okay. So now this is the viewing the sequencing files. Okay. So now if you have a sample file, say this is the FASTA files that we are getting. So we can download any FASTA file from the internet, and then we can save it and then we can use. So this is just a command that I have written. Okay, so we are going to look from this. So we had the file hello. So we'll do vi wait. So the file was 2.txt in which I have written. So I will just edit that file once again. So I have already only written hello. Okay. So I will add some more things like hello, hi, how are you? And I'll just add some characters. Okay. And then like I will just close it. So now I will again, that was just I'm writing inside the file. So now I am going to again visualize that file 2.txt. So now you can see that all of this content I've written inside my file. So cat is going to show you the entire file. Okay. It is going to show you the entire file that is present. There are sometimes that your file has 1,000 pages. You can't show all the 1,000 thing on a visualize is head 2.txt.
Anshu, sorry to interrupt you. We couldn't hear you, actually.
Actual
We
parts
heard,
of the
Anshu, can you hear me?
yeah.
Yeah. Actually, we lost you a few seconds back when you were speaking about head command, before the head command.
Okay.
You were saying that if we want to see 1,000 lines, till that we hear.
Okay. So if the cat command basically shows the entire file. Okay, if you have a file that has 1,000 lines, so when you do use the cat command, you will see that the 1,000 lines will come here. The terminal will keep on going because the file is very large. You don't want to do that. So there is a command called as a head command and a tail command. As the name suggests, head, it will show the start of the file. Tail, it will show the end of the file. So if I do head.txt, as this file is small, so that is why it is only showing this. So in 1 second, I will just edit it once so that I can show you how it works. Sorry. Okay. So when I do now cat2.txt, you can see that it is so big file. Okay. So from here it starts hello, how are you? And it goes so down. So there are chances that the files can be much more longer. So now I just want to see the initial part of this file. So I'll do head2.txt. So you can see it is only showing me this much, even though the file is this much long, but it is only going to show the initial few lines. That is what the head command does. And when I'll do tail, you can see that it will not show the start part. It will only show few lines from the bottom. So these are the few lines that are shown. So head and the tail command is used to basically see the file content that is present in the start and in the end. So these commands are generally used when you have a very large file. When you have small files, you can directly use the command cat. Is it clear? Are you guys able to understand? Can I get a thumbs up? Okay.
Anshu, one small request. Can you also say us how to edit the text file?
Oh, yes. This is a VI text editor. Majorly what you use is like it depends on what these people are comfortable in. So most probably what I do is generally open the text editor in the notebook or in the notepad and edit in that as well. So this is just for a slight like VI editor. Um, VI editor is going to be a little like it will take some time to make people understand. So what I can suggest is if you're creating a file, just double tap on it and then you can open it in a notepad and you can edit it there directly. Is it clear, everyone?
If possible, can you also show that in the folder? Because I think
Okay,
you might
yeah.
not
Once,
understand.
yeah, okay. So now you guys understood this, right? The commands. Now I'll show you how you can edit these files. So this is my directory path. So when I open my files, I can directly go into it and I can copy my path here and I can open it. Sorry. Okay. So if we create a file here. So like right now I'm having some problem with my system. Like I'm not able to access the thing. So maybe I can take this next time. So right now I'll just share that you can just open the text files in the notepad. Okay. So if you have to create a file, 1 second, you can directly create a text document. Okay, so you can directly create a text document. Say it, I'll name it as 1.txt. So this you will be doing in that in your Linux system, and wherever that data is getting stored, you are going to open it. So now you just double click on the text file. You can see that a text notepad is getting open. So whatever you want to edit in that text file, you can write as hi, hello, whatever you want to write here, control S, save it, and then you can open and you can understand it in your Linux environment, that is in your terminal. So if you want to edit any file, you can directly open the notepad. You can put your file, you can put the write on the text there, and you can understand, and then you can run it in the, you can whenever you are opening this folder. Okay, so in this particular directory, so this is going to be available with you offline in the system as well. So you can just double click on this and then you can open. I'll show this as well in the next class once. Like I'll just fix my paths and all in my system. It might take some time. You have to cover a lot of things in the session. So once I'll figure that out, I'll show that tomorrow, that how you can edit in the notepad. Till now, is it clear how the basic commands are running? Okay. So now there are certain things that is another like important. So whenever you have to compress a file, so if there is a very large file and if you want to send a file somewhere, you have a very normal option in Windows where you just right click on the file and you can get a not a compressed file option. But in this particular thing, that is in Linux, you have commands to do that as well. So you can directly write a prompt on the terminal and you can get your file zipped. So now you have this 2.txt, right? So when you have this 2.txt file, you want to zip it or you want to compress it. So you use the command z zip and you write the name file. But this is a text file. Do you guys think that this should get zipped or not? Do we get a folder zip or do we get a file zip? Do you think that this command should work? Can I zip a text file?
Guys, can anyone unmute and answer this? The question is whether you can compress a text file.
I think there will be a cross or a tick emoji. You guys can just send that as well.
Session being silent, I now go to a state whether these guys are understanding or not.
Yeah, that is my
I
major
mean,
concern.
they're active or not. I am in dilemma
Yeah.
with this guy for active.
Like I'm thinking that am I able to make them understand what I'm trying to say for like last 1 hour?
Yes. Any volunteer would like to unmute and say, or I should pick the name randomly.
I think name picking should happen now.
Yes. And should
We
you like
guys
to
gave
take
you,
a call? Or-
yeah. I'll say a name. We guys gave you enough chance to speak on your own, so I'll ask Maria to tell. Like, is it possible to zip a text file or not? Like, you just have to give a yes and a no answer. It's fine if it is wrong.
Yes, ma'am, we can like, ma'am, we can zip files.
No,
You
the
can
question
zip-
is we can zip file, but the question is whether we can zip a text file or not.
Ma'am, I'm not sure.
You're not
Okay,
sure.
it's okay. Yeah.
Varsha?
So,
No, let's take one more call.
yeah.
Varsha?
Yes, ma'am.
Yes. Hope you got the question.
Ma'am, I'm thinking, ma'am.
Thinking. Okay. Right. And so let's proceed.
Okay, so see, now we'll see if we can zip a text file or not. So when we try to zip 2.txt, you can see that it got zipped, right? So yes, we can zip a text file as well. So this was just a trick question that I wanted to ask you guys. Now you can see, right? We had 2.txt and now it got zipped. So now we have a 2.txt.gz. So gz is a symbol where we understand that, okay, the file is getting zipped. So similarly, when you have to like decompress a file, then you use a command gun zip. Okay, that is you want to reverse it. So we'll put gunzip 2.txt. Now you can see the file is reversed back to the normal format. So it is no more a zipped version. Okay. And then now there is another command that is zcat. Okay, cat command is used for visualizing the visualizing the normal file. So if you want to visualize a zipped file, like you directly can't visualize a zip file, you have to unzip it and then only visualize it. So, but if you have to visualize a zip file directly, see you have a zipped file, right? 2.txt.gz. So now we will use a command zcat. So zcat can directly show you the what is present in the compressed file. So now if I use the command cat, you can see that it is not going to show anything. You saw some random symbols came because it is a compressed file. It cannot be read by a normal cat command. So zcat is able to read that text file and understand and print it onto the screen. So these are the basic commands that were basically used for working and working with the compressed files. Okay. So now there are certain other commands like this was the zipping commands. Okay, so now there are certain other commands, that is pipe and redirection. So if you want to combine two commands together, then you can use a symbol that is a pipe symbol. So this symbol what I am showing right now, one second. So this is a pipe symbol. So you write some command here and then you put a pipe and then you write some command here like this. This is just an overview. So if you do like this, then this pipe is going to basically connect these two commands. Okay, so what you want to do is you want to visualize say 2.txt file. Okay, cat2.txt, and then you put a pipe, and another thing is you want to do grep. So I will explain what is grep as well. So you do grep and we'll see for a symbol. Okay, so you put grep, then okay, one second. You put grep, then you choose, and then you put this symbol. So this symbol is basically that you want us, like you want to visualize from this particular file. One second, guys, my system is hanging a little. Okay, so when you have to combine two commands, so you basically write one command, that is cat2.txt you have written here, and then you include it with another command, that is like a grep command or something. Okay. Okay, I apologize for that. Something is wrong with my system. Okay. So now for the thing, I'll just try once again, opening it again. So till the time it is opening, I would like you to understand that the commands, this pipe command is basically used. Okay, it's there. Okay, so we were working in this particular folder. Okay, so now we moved on to a text file folder. Okay, so now first we'll decompress this file because we had a compressed file. So we will use the command gunzip to basically unzip the file, and then this file is now unzipped. So now we were learning how to combine the commands. So now first I'll do is cat2.txt, Now I want to visualize whatever content is there. And then I would like to put a pipe. Pipe is going to combine two commands. So now I'm going to use a grep command that is going to tell. Okay, so the grep command is not working for me. So I'll, what you can say. So I'll put a cat2.txt, and then I will just like to list all the files. So when you do cat2.txt and list, so basically it is going to list the list all the files, and it doesn't show the cat2.txt because the command 1 is not compatible with command 2. So this command is basically used to combine the two different commands that you are working with. So grep command, if I have to write, so grep command basically shows, I want to demonstrate the grep command, but it is not working for my system. So grep command is basically going to look in the file. So when you do cat2.txt, so if you want to search how many V's are there, so you put grep along with V here. So in this command, so when you put a V there, then you can see that only it will print those line that has V inside it. So grep is basically a selective command where you can choose and select out certain things from the file that you are looking into. So after that, there is a redirection file. That is, you save the some output of a command into a file. So say you ran any command. So this commands I'm going to explain much more in detail when we are going to work with our RNA-seq data. So if you are going to say, if you generated a file, like if you ran a command where you are writing something in a file. So you're writing, say you're writing some content, say some hello content in a file. So we can try running this command if this works. Otherwise, I will move this further with you in that thing. Okay, so this will not work. So when we are going to write something into a file, so you ran a command where you did RNA read mapping. Okay, after mapping, the data got generated, and that generated data you need to store in a particular file. So that particular file, you instead of writing like a separate command for that file, you can just directly put a greater than symbol, and that entire content what is getting generated will be saved into a text file. And similarly, you can append all of that notes or whatever it is into your file. So these commands, you can just understand and look into it here. When we move on to the RNA-seq data, we are going to perform this because we need to run certain commands and get certain outputs on the terminal that you can write into your text files. So next is a FASTQ file. So FASTQ file is basically that we'll be working with. Okay. So whatever data that we are getting from, okay, before starting with this, are all the Linux basic commands clear? The people who had their systems, are they able to like run the same commands? Only one thumbs up. Other people? The people who had their Linux systems, are you able to run these commands? Did you guys try running like even a one command?
Yes, ma'am.
Okay. So you guys can practice this once again, and whoever doesn't have the system right now, they can go later and like do it again. So these were the basic commands that I wanted like to go through. So this covers everything, whatever basics you need to know about Linux before you work in a RNA-seq environment. So now we'll just understand the FASTQ file like in a basic and a crisp manner. So this is how generally a FASTQC file looks like. Okay, so basically this is a language which we don't understand, but the computer understands. Okay, so that is why it is written like this. So the first line is written as @sequence ID. So you will have a sequence ID which is like followed by a @ symbol. So this identifies that this is a line identifier. Okay, a new line is getting started from here. The line 2 will have all your DNA sequences, that is the bases A, T, G, C, the entire sequence that is sequenced with the help of a sequencer. Line 3 acts as a separator, that is you will generally separate it from these lines. There's a break between these two. And then you have the quality scores. So these quality scores are encrypted and these are encoded. Okay, this is not directly what it looks like. So each quality score is represented by a number here, or a number, or you can say an alphabet. Okay, this alphabet is read by the system, and for that particular alphabet, a quality score is given. So these are very much standard. If you have I, it will correspond to a particular Phred score. If you have another alphabet like A, it will correspond to a particular Phred score. So these Phred scores are the what that we analyze during the quality that we check during the fast QC analysis. That is the first analysis or the first step that we take for our RNA-seq analysis. So this is a basic thing how our fast QC file looks. Similar way, it is going to be multiple columns and multiple rows and much more longer sequences. This is a very small sequence. It is going to be a much more longer sequence than this. So this is how a normal fast QC file basically look. Okay. So now, so now we have come to the tools for the system. So what and all tools that are required for our analysis. So what you can do is, so these are the commands. So first of all, you need to update your Linux environment, that is sudo apt update. So what you'll do is, in your Linux environment, you'll do just sudo apt update. So you will do the sudo apt update. You guys can do simultaneously as well with me. It is going to update your Linux system. You need to write the password that you have set for your system. So you enter that and it is going to update the entire system and entire Linux environment. Whatever updates are required, it is going to update all of it. So before doing any analysis, you generally update your system so that it works more efficiently. Okay. So after working all this, so now you require to install, now you have to install certain tools. So those tools include FastQC, FastP, Bowtie 2, SAM tools. So these are the tools that we'll be using and you need to install. So once the session is over, I'm going to upload this presentation. You guys take a reference of it and then you have to follow these steps and you have to install all the tools that I've mentioned in the presentation. So what you have to do is simply see, you can see that whatever it was there, it got updated. These are the some things that needs to be upgraded and are a problem with my system. So we'll just ignore that. So now, okay. So now what we will do is we are going to install the tools. So first we'll do is sudo apt install FastQC. Okay, so first I'll go in my directory that I've made for you guys. So that is RNA-seq 2. All the work we'll be doing in this directory. So we'll install sudo apt install the FastQC. That is our first command. Okay. Okay, so you have to write this command. It will not show like this for you. It is going to install the entire FastQC app inside this Linux environment. So this tool will get installed. Similarly, you have to write FastQC, FastP, Bowtie, and SAM tools. So you just have to copy this entire command from here. Like you'll copy this entire command, you'll paste it there. All the tools will get installed automatically once you have put it into your Linux or your terminal. Okay.
Anshu, one small doubt to me here, whether they're going to prefetch it's using the SRA toolkit or what?
No, we are just installing the tool, so we can just directly do it by sudo apt install.
No, I'm just asking whether for our project we're going to prefetch the sequence. I'm asking whether we're going to prefetch from SRA or what, because
no, I'm going to give a small data set. Because of the computational system handling, I'm going to teach them how they can take the data from SRA, but I'm going to give a data set on which everybody is going to work alongside.
okay.
Yeah.
But when it comes to offline students, they're going to prefetch it, or for them also you're going to give the data?
I'm going to give the same data they have to reproduce for their study. So I'll share it on the Drive link as well.
Okay.
Okay. Okay, so this is the installation part. Next is what and all these tools are functioning. So FastQC tool is going to be used for analyzing and assessment, the quality assessment and the raw sequencing reads. FastP is used for trimming the sequence, that from whatever place the quality is bad, you can trim those sequence from there by using FastP. Bowtie is an alignment tool, that is you have a reference sequence to which your transcriptome, that is the reads that have come, will get aligned. And that alignment will be analyzed further. So this analysis is done by Bowtie. HiSET is also an analysis tool, but it is a generally preferred one in RNA-seq because it is aware of the transsplicing events. That is, it knows that the entire genome cannot be mapped. There will be certain introns and exons, and that should be mapped accordingly. Then SAM tools. SAM tools is basically used for conversions, like conversion of SAM to BAM or BAM to SAM, or indexing or sorting the files. For that, we use this. And then R we use for the installation. And then for R, in R, we are going to do the further analysis where we are going to generate all the statistical charts. So these tools, the initial, we are not going to install HiSat 2 because we are not using it. So these 3 and this one, 4, these 4 tools should be installed in your terminal. And R, we are going to use an interface. That is, we are going to install RStudio, and that we are going to use. So after you have installed all these apps, you can use these commands to check the versions of what app is in what is the version of your particular tool that you have installed. This just acts as a verification. If your tool is installed, then only this command will show a result. If your tool isn't installed, then this command will show some error or something. So now how to install R. Okay, so for R, I'll also show you how to install. So I've put a website here. So you have to go to the website. You have to put it here. Now it is going to open a web page. From that, you are going to install your
Ms.
R for
Anshu,
your.
before proceeding with that, can you please demonstrate one command here, one tool here? Maybe you can just download one and show us how does the version looks like. Just one.
The thing is that there is some error happening from my end. I don't understand this. Like it
No
is not
problem.
installed.
We post
Yeah.
the session. You can share the
Yeah,
screenshot.
yeah. Yes, I can share a screenshot. I tried to install, but it is denying me the installation. So I need to decode, like I need to figure out what to do with this. So it might take a little time. So after the session, I'll even record a video of how me installing it, and I will share it on the drive so these people can understand how to install. Yeah.
Thank you.
So for that, for our installation, I have mentioned a website here that is this Cloud R project. So you copy this website, you directly go and put it in the browser. You will this particular web page will open. So you have to click download R for Windows, and it will start. It will take you to a different web page, and then you from there you can start downloading. So you install R for the first time. So you install here. It will keep on showing you the steps, and you install R, and then R is done in your system. So once R is installed, then you can check the version from your terminal, and you can check like run the installer and all. It will give a proper installation guide. You can go through it and you can install it properly. It's a long process that you need to take some time and follow. If you guys face any problem, you can directly contact me. You can tell me, okay, ma'am, here and all, we faced a problem. So I'll be helping you guys throughout. That is not a problem at all. Okay. So once R is installed, then the next step is installing the RStudio. So similarly for installing RStudio, I've also given a link. So you just need to copy it. You paste it in the web browser.
I think I'm sure facing some issue, right? We couldn't hear it. Ms. Anshu, can you please unmute yourself?
Okay. So I'm saying that for RStudio, for the installation of RStudio, I have given a link here. Once you are done installing R, then only you should move to the RStudio step. So you put the RStudio link into the web browser. So when you put it here, it will show a page automatically where there will be option given where that install RStudio for Windows. Okay. So you directly install the RStudio for Windows, and then you're good to go. So now you have RStudio installed in your system. You might need to do some configurations. And if like for each system, there are different configurations. Okay. So even if I show you right now what to do, that might not be the same for you, because for us, like if 40 students do, there might be 40 different situations that come up. Okay. So from this link, you can download the RStudio desktop for Windows. And once you have downloaded it from here, then you can start working and configure it, and then you can start working with it. So So once we are done, so I will just show you the web page once. So once we are done with our studio download and installation, so what we will do is we will open it. So it should look like this. Okay, so you will have a R Studio app in your Windows. So once you open it, you will be able to see an entire app of R, which will have certain terminals. So let it open. I will show you how does it look like. Okay, so this is basically it has its own terminal as well, and it has a place where you write the codes, where you can see the results, where you can see the tables. So everything is combined in one. So it is a better representation and a better way to use the like R. In terminal also you can download and install and use R, but that becomes a little messy because you have to write all the commands on terminal. Whereas in R Studio, there is a designated area for you to write commands and a designated space to see the results. So you can anytime edit and open and see. So till the time it's opening, we'll move on to the presentation. So it might take a little time to open because it's a little computationally heavy because we'll able to do it. So wait a second. After presentation, maybe I can open it and show it to you, show this to you guys. Are you guys familiar with R Studio? Have you guys ever used it before? Any one of you?
Yes, ma'am.
Okay, so if you are familiar with it, then you will be familiar with the GUI as well. So we'll look into it once. So once our R is downloaded and we have an R Studio, so now what we are going to do is we are going to install our bioconductor. So now we are going to install certain packages in R. So this is the command that you need to run. Once R is there, you open the R environment, and after you open it, you can see a place where you can write the codes. Where you write the codes, you just put this install packages BIOC manager. So once you put this, it will start installing. So it will install a BIOC manager in which you can further use to install all the packages that is required. So these commands are used to install all the packages. So you just need to put the entire thing, put it into the system and run all these commands. So after finishing all of this, you will have all these libraries: rsubread, edgeR, lemma, ggplot, and pheatmap. All these libraries are required to work in R for the analysis. After all the libraries are installed, we will generally load the libraries. This is the command to load the library. You write library followed by the name of the library that you need to load. So now we'll just talk about these libraries. So first we have BIOC manager. So BIOC manager, it acts as a manager of all the packages that is present. So it installs the bioconductor package. From that package, whatever library we need, we can install regarding with help of this bioconductor. So rsubread is basically used to do the feature count. That is, it will count the number of reads and samples and form a count matrix. This count matrix is further given to the further differential tool analysis, that is edgeR, lemma, and all, which does the differential gene expression analysis. So it will normalize the data. It will fit to a model. We will read about this in much more detail when we're going to perform this. First, we'll study the theoretical part and then we'll perform it. So it fits the normal data. That is, it will normalize, bring it down to a normal scale, calculate the log fold change, p-value, all of that. And then based on the statistic models and statistic analysis, it will create and analyze all of this. And then it will give us the graphs. So these graphs are plotted with the help of the library ggplot, that is the volcano plot, and heat map is plotted with the help of a library pheatmap. So in order for you to plot these graphs, you should have these libraries up and running in your R environment. So ggplot helps to plot other bar charts, scatter plots as well. It is a normal plotting library. pheatmap is very specific for plotting heat maps. So this is the information of all the tools that you're going to be installing and all the libraries inside R. So now, where are all these packages and libraries are getting used? So initially we had FastQ files, right? Those FastQ files are going to be analyzed by the FastQC tool that we'll be installing in the terminal. In the terminal itself, the bowtie command that we will be installing will be used for mapping into the reference genome. Now that is going to construct the SAM files. So we need to do the indexing and sorting of the SAM and BAM files that are produced by the use of SAM tools, or bowtie 2 can also do the same. Now we will move into R. So now we have these files generated in the terminal. So now from the terminal, we need to move into R, where R is going to read this BAM file and it's going to give us the feature count. So this feature count acts as a matrix that is given to HR and Limba. These are the tools that does the statistical analysis and the differential gene expression. So it runs, it normalizes the data and gives the data for the differential expression. So now this differential express data that we have got, now we can plot it using two tools. One is ggplot2 and one is pheatmap. And then you can visualize the plots that are getting generated. So the information that is given by this flowchart is basically that where at what step in the RNA-seq analysis you are using these particular libraries and tools that you have installed in your system. So now we'll see how these plots basically look. So when we are doing an RNA-seq analysis, so I told you that we use the ggplot. ggplot2 is a very good visualization tool and it generates these plots. So it apart from Volcano plot, it can generate any plot that is like you need to generate in R, but Volcano plot is what we generally generate in RNA-seq. So it generally looks like this. So we'll also generate a very similar image with a data set. You can see that there's a log fold change here and there is a log FDR here. So FDR is also a rate. We'll study about that as well. So basically from here, what you have to understand is that this is a Volcano plot. You can see whatever is in blue and whatever is in red. So according to your scale, you can check and you can see that this is upregulated genes, this is downregulated genes. And the genes that are in gray, they are not much differentially expressed. They're having the same expression in the disease condition and in the normal condition. So you can only, you have to only find the genes that are very highly expressed and which are very low expressed. So similarly, heat map will give a heat map visualization. So what you can see from this heat map is you have been given a scale here, right? So 4, 2, 0, -2, -4. So the blocks that have the red value, that shows that they are highly expressed. The ones that shows blue value, they are showed that, okay, they're very less expressed. So wherever there is red, you can understand that those genes are very highly expressed. Wherever it is blue, it shows that those genes are not very particularly highly expressed. So this is just a proper, like a bigger picture. You can see that all the red blocks, those can be considered as high expression genes. The blue ones, more dark the blue is, more or less the gene is expressed. So the transition from blue to red shows that the expression of gene is increasing as the transition is happening. Similarly here, the red also shows very high expression, the cells, and the blue also shows very low expression. So this is what we are comparing and what we need. So this is what we analyze after we perform our analysis, that is an RNA-seq analysis. So after that RNA-seq analysis, you can understand the differentially expressed genes by the means of these two plots, that is a Volcano plot and a heat map. So now from the raw sequencing to the biological discovery, so basically this is the final pathway that we are taking entirely. This is to summarize the entire thing from the start till the end. So you will have a FASTQ file, you will perform a quality control, that is the FASTQC. Then if required, you can do trimming. We generally don't do that much trimming in the RNA-seq analysis because the adapters are generally removed after the sequencing itself, and those adapters are something that we know the sequence of. So that then alignment we perform by using bow tie and HiSat 2. So bow tie is like a normal one. HiSat 2 is one that also considers the splicing events. Then after alignment, SAM and BAM files are created. These SAM and BAM files are indexed, sorted, and all of that things are performed. And now these SAM and BAM files are given as an input for the feature count. From this analysis, we can understand how many reads are aligning to which part and I'm sorry. So what are the genes, what are the samples, how many reads are aligning to it, all of them are included in this feature count. This feature count is converted into a count matrix, that is a sample and gene. Now this count matrix acts as a raw read count for the analysis of the differential expression. So this raw count is fed into HR or DESeq2 in R. And then they calculate the differentially expressed genes, normalize the data, and then give us the data in columns of the upregulated genes and the downregulated genes. Now for those upregulated and downregulated genes, we plot a Volcano plot or a heat map or a PCA. So for our analysis, we are going to plot all these three plots: Volcano plot, PCA, and heat map for our analysis. The analysis. After we get this, now we upload those differentially expressed genes into the enrichment websites like GEO and KEGG and understand that, okay, which gene is corresponding to what function in a particular body. That is why we can understand that what protein is elevated or what protein is decreased in a diseased or a normal condition. So this is the basic and the conclusion of whatever we studied in our particular study. So we studied about the Linux fundamentals, the NGS and RNA-seq workflows, and whatever the tools are required for working in the terminal. What are the outcomes? That is, we get ggplots to generate volcano plots, the heat maps for interpretation, the PCA plots, and then this and all plots are generally produced by R. So you are producing and analyzing all this data in R by the differential expression analysis tools using EdgeR and DEC2. So this was a basic understanding of all the tools. So this session was basically for you guys to understand and install. So I hope it is clear what and all tools are required. So before the next session, you guys should install all these packages. Basically, first you need to install RStudio, and all these commands. That is all the list, the entire list of this command should be installed except for HiSet2. All of this should be installed in your systems. So this is and all is required when we start with a practical exposure to the RNA-seq. So all these tools will be required. So install all of this and keep your systems ready so that you can work directly. I will post a video as well in showing one or the two tools getting installed, like how do you write and how it is generally seen, so that it becomes easy for you guys to understand as well. And some people also told that you guys have already some understanding and some exposure to RStudio. So you guys, I hope you guys can install that as well. And if you guys have any problem, you guys can just post your doubts, and as soon as it comes to me, I'll clear them as well. So for R, I have given everything, all the commands. You go to those websites, you install the packages, whatever I've shown you in the session right now. And I think that will be enough. So after all of this is done, then we can move to the next sessions where we'll be analyzing all this data in much more detail. Like we'll be using these tools in like hands-on to understand and to analyze the data. Is it clear? Is it clear for everyone?
I think that should be clear. We got few thumbs up.
Yeah.
All right.
Yeah. Okay.
I think we have came into end of the session, right?
Yes. Yes.
Okay. Fine. Holy Cross girls, I think this is clear because you're going to install this at your system. If you have any doubts, any clarification, please post in our WhatsApp group. We will try to sort it out. And when it comes to hybrid session, students from Anna University and RSC, please don't worry about installation part. Kindly observe the session. All this installation we can do in our lab. Okay. So please don't worry about this now, but please observe the session. Is that fine? Anna University Chennai, guys. Please give thumbs up. All right. How about Gokul? Are you clear? Fine. All right. Holy Cross girls, please make a clean attention. You need to install all these things, all these tools. If you have any issues, please keep us posting. If you have any doubts, please let us know. I'm requesting it once again because this plays a fundamental for your entire project. So please pay keen attention in installing all these things. Yeah, thank you. Anshu, do you want to say them anything for next session?
The only thing is that they should practice these commands that I have just explained.
Okay.
Because these are the basics. Later, I might ask
your voice.
to show also as well, like the online people that have the access to their system. I might ask as well that you guys should share screen and show the commands running. Okay, just mentioned in today's session. Just revise that once properly. You should be like thorough with that.
Okay, let's have one homework now. Okay, what you guys can do is you can practice all these commands. Okay, in Linux environment, after practicing all these things, once you type history, you can see all the previous commands which you have got. Okay, just take a screenshot of it and post it in WhatsApp group. Okay, hope this is clear. Just run all these commands one by one. Don't close the terminal. Type a command history, which will show all the previously used command. So take a screenshot and share it in our WhatsApp group. This is for everyone. For those who are attending it online, offline, whoever. This is common for all. And we need it by tomorrow, 5 p.m. All right. I think it's time to wind up as you guys have other commitment by this time. If you have any doubts, I'm requesting you all to post it in group once again. Thank you all. You will be receiving this presentation. When you will be posting this in Drive, Anshu?
I'll post it right away. They can access. Yeah, they can access another 5 or 10 minutes. I'll just post it right
All
away.
This presentation slides to which the commands is included will be posted to you in the Drive. I think it will be done in another 10 to 15 minutes. And session recordings you will be receiving at 10 p.m. today. Okay. Kindly practice it. Thank you all. And one request, please fill the feedback form. Feedback form also act as your attendance form. So kindly fill it out. Let's meet tomorrow at 6 p.m. Thank you all. Thank you, Anshu.
Thank you all. Thank you, everyone. Bye.
Thank you, ma'am.